Hello everyone, today we are going to learn about ovarian culture and its applications, which mainly include the following contents: ovarian structure and function, in vitro methods for studying ovarian function, method and observation of ovarian culture, cautions, applications, and limitations of ovarian culture. Firstly, let's see the structure and function of the ovary. Ovary is an important female reproductive endocrine organ with two main functions, including producing oocytes and secreting female hormones. Adult ovary contains follicles at different stages of development. Follicle is the basic structure and functional unit of the ovary. It is composed of an oocyte and surrounding follicular cells. According to the developmental stages, follicles can be divided into primordial, primary, secondary, entrant, and pre-ovulatory follicles. Cyclic regulation of ovarian function is important for female reproductive health. The cyclic changes of female reproductive system are mainly regulated by the secretion of reproductive hormones from the hypothalamic, pituitary, ovarian axis, and the regulation of gonadotropins secreted by the pituitary gland. The ovary exhibits cyclic changes in follicle recruitment, growth, maturation, ovulation, and corpus luteum formation. In addition to the regulation of reproductive endocrine axis, there are many factors affecting ovarian function in vivo, such as a variety of endocrine hormones and growth factors inside and outside the ovary. The location of the ovary and the complexity of the internal environment make it difficult to manipulate the ovary and study its function. Therefore, it is crucial to explore methods for in vitro studies of ovarian function. In vitro culture is widely used in ovarian functional study due to its advantages of simple manipulation, few influencing factors, short culture time, easy operation and intervention. Commonly used in vitro methods include ovarian culture, follicular culture, granular cell culture, interstitial cell culture, oocyte and early embryo culture, etc. The proper application of various in vitro methods is helpful to study ovarian function and review the pathogenesis of ovarian disease, such as premature ovarian insufficiency. In this section, we will focus on ovarian culture, which can be divided into embryonic ovarian culture, neonatal ovarian culture, and adult ovarian tissue culture according to the purpose of the study. The specific methods and precautions for in vitro ovarian culture are as follows. Because of the small size, suspension culture of intact ovaries can be applied to the ovaries of mice from embryonic day 13.5 to 4 days after parturition. Nutrients and oxygen can easily penetrate into the intact ovarian tissue, and the longest cultured ovary can survive for about 2 weeks. Adult mouse or human ovaries, because of their large sizes, need to be prepared into tissue sections or divided into small pieces before they are cultured in vitro. The commonly used culture medium is DMEM F12 supplemented with 1 microgram per milliliter bovine serum albumin, 1 milligram per milliliter albumax2 lipid-rich BSA, 5% ITS, 100 micromolar ascorbic acid, and 1% penicillin and streptomycin. Finally, the ovary can be cultured in a tissue incubator with 5% carbon dioxide and 37 degrees centigrade. During the culture process, the ovaries can be observed to become large in size and flat in shape. The follicular formation, activation, and early development can be observed after section staining. For example, secondary follicles containing 2 layers of granular cells can be observed in PD4 mice ovaries after 2 weeks cultured in vitro. The following cautions in ovarian culture require special attention. First, avoid disrupting ovarian structure and operate as quickly as possible during ovarian dissection. Avoid air bubbles when adding medium to the inside and outside of the culture insert. Avoid adhesion between ovaries when placing them in the culture insert. Avoid excess medium inside the culture insert during incubation. Applications of ovarian culture. Ovarian culture can be used to study ovarian development, early follicular development, and homeostasis and secretion. Superficially, culturing of embryonic mouse ovary can be used for the study of early ovarian development and germ cell development. Culturing of neonatal mouse ovary can be used to study the process of primordial follicle assembly and formation. Culturing of PD3 to PD4 mouse ovary can be used to study primordial follicle activation process. Culturing of adult mouse or human ovarian tissue can also be used to study primordial follicle activation. In addition, culturing of ovarian tissue has also been used to assess the female reproductive toxicity. Finally, the limitations of ovarian culture cannot be ignored. The duration of in vitro ovarian culture is too short to complete the whole process of follicular development. Especially in postnatal mice, the follicles cannot develop beyond the secondary follicle stage. In addition, due to the lack of cyclic regulation of reproductive endocrine axis, in vitro ovarian culture cannot fully reflect the physiological process of follicular development in vivo. Furthermore, in vitro ovarian culture requires various components of the culture medium, which may affect the survival and the developmental potential of the ovaries. Therefore, more appropriate ovarian culture conditions need to be further explored. To summarize, in this section, we have learned the methods, applications, and the limitations of ovarian culture. You can choose and apply them according to the purpose of your study. Thank you for your attention.